Good evening, Doctor, and to all other friends. We are from Group 1, and our topic presentation on today is Apoptosis in C.Elegans. Let me introduce my group members. My name is Linkeshwaran Kanagasiban, and my group members are Durkashini Ravichandran, Lang Suk Mei, Liang Jun Hee, and Karishma Sadanandan. We took question 1 to present on today, and the question is: Apoptosis is an important event that happens during embryogenesis. Present an example of apoptosis that happens during C. elegans embryogenesis. Next, we discuss about the process of embryogenesis in C. elegans. During embryogenesis, the zygotes exhibits rotational holoblastic cleavage. Each asymmetrical division produces one stem cell and one founder cell. Physically asymmetric division of the C. elegans zygotes ensures invariably successful embryogenesis. Our main focus on today is apoptosis in C. elegans during embryogenesis. Apoptosis is defined as a gene-directed cellular self-destruction program that serves biologically meaningful functions to organisms. In C. elegans, apoptosis is normal component of growth. During embryogenesis, 131 cells undergo apoptosis out of 1090 cells of somatic cell. Let's take a look on C. elegans. What is C. elegans? C. elegans or Cynohabditis elegans is a free-living transparent nematode that about 1 mm in length that lives in temperate soil environment. These C. elegans are used as model organism to study about human diseases. Most of these nematodes are hermaphrodites and a few are males.
C. elegans as model organism. First, it has the psychological properties of an animal and has a fast life cycle. Next, C. elegans is one of the simplest organisms with a nervous system. In the hermaphrodites, this comprises 302 neurons whose pattern of connectivity has been completely mapped out. Third, it's been used to assist in novel human gene identification, human disease and also human system. This is because C. elegans proteome was used as an alignment template to assist in novel human gene identification from the human ESD nucleotide database. Among the available 18,452 C. elegans protein sequences, the result indicates that at least 83% sequences of C. elegans proteome has human homologous genes, with 7,954 records of C. elegans protein matching known human gene transcripts. Only 11% or less of C. elegans proteome contains pneumatode-specific genes. Thus, C. elegans are used in identification of more than 24 genes that are important for the specification of which cell should live or die, the activation of the suicide program, and the dismantling and removal of dying cells. There are various cell death specification happens in C. elegans. Previously, we have learned NSM and HNS formation. The other cell death specification is tail spike cell, NSM sister, P11.AAP, and CM neurons. In all these processes, there are four main genes that function as cell death promoting genes, which are EGL1, CED9, CED4, and CED3.
The example of apoptotic event that happens during C. elegans is during hermaphrodite specific cell death, CEM, which means cephalic male. So a brief introduction about the CEM is: the male is protected from the cell death due to the transcriptional upregulation of CEH30 by UNC86 in the absence of TRA1A, which antagonizes the activity of UNC86. The direct target of CEH30 or UNC37 reprocessor is remained to be identified.
CEM cell death requires all four core cell death genes, which are EGL1, CED9, CED4, and CED3, as similar in the germline and tail spike cell. CEM cell death regulation appears to require transcriptional activation of the CED3 caspase gene. Although EGL1 expression is still induced in CEM neurons, this induction is not always sufficient to promote CEM death.
Hi everyone, now I will talk about EGL1. EGL1 is the first BH3 only gene. It is a key activator of apoptotic cell death in C. elegans. EGL1 activates programmed cell death by binding to and directly inhibiting the activity of CED9 and releasing the cell death activator CED4 from a CED9-CED4 containing protein complex. Loss of function EGL1 mutation prevents most but not all somatic programmed cell death. Next, I will talk about CED3 gene. CED3 gene is one of the major protein components of the programmed cell death PCD pathway for C. elegans. It acts as the core apoptotic cell death executioner. It acts in early responses to neuronal injury to promote rapid regenerations of dissociated axons. CD3 helps in the development and growth of the organism. Without apoptosis, the cells that have been damaged or aged will not be able to be replaced with newer, healthier cells, thus inducing growth. Another gene I would like to talk about is CH30. CH30 is required for the sexually dimorphic survival of the male-specific CEM sensory neurons. CH30 encodes a cell type-specific anti-apoptotic homeodomain transcription factor. Anti-apoptotic function of CH30 acts within the CEM neurons to promote their survival independently of both EGL1 and CED9. CH30 protein is required to repress EGL1 and CED3 transcription in the CEMs of masculinized hermaphrodites, thereby causing their survival.
To the transcriptional upregulation of both EGL1 BH3 only and also CED3 caspase. During the development of C. elegans hermaphrodite, exactly 131 cells are programmed to die. The majority of the cells die around 30 minutes after being generated. Furthermore, in these cells, the transcriptional activation of BH3 only gene EGL1 is thought to be sufficient for the activation of central cell death machinery and also apoptosis induction. Specifically, the BH3 only protein EGL1 can bind to the CED9, thereby causing CED4 released from CED9 and CED4 dependent pro-CED3 maturation. Interestingly, the recent data suggests that CED3 gene is not actually transcriptionally active in the most of the 131 cells that are programmed to die during development. So this observation suggests that the pro-CED3 protein present in these cells is derived from their progenitors. It also suggests that once activated, the amount of pro-CED3 protein inherited from the progenitors is generally sufficient for apoptosis induction. The four male-specific cephalic companion neurons (CEMs) are generated around 320 minutes after the first cell division. In males, the CEMs survive and differentiate into ciliated sensory neurons implicated in mating behavior. So in hermaphrodites, however, the CEMs undergo apoptosis around 150 minutes after being generated, approximately 470 minutes after the first cell division. So as in the case for most of the cells that are programmed to die during the development, the death of the CEMs is dependent on the genes EGL1, CED4 and also CED3.
The death of CEM also dependent on gene TRA1. Gene TRA1 encodes the terminal global regulator of somatic sexual fate that acts to promote female development. TRA1 protein, a GLI-like transcription factor that causes the CEM in hermaphrodite to undergo apoptosis by directly repressing the transcription of gene CH30, which encodes a bar homeodomain transcription factor that acts to block the death of the CEM in male. We now present evidence that CEH30 blocks the death of the CEM by repressing the transcription of both the EGR1 and CED3 gene. Furthermore, we identified three genes, UNC86, IRS1, and UNC132, which encode a PAL homeodomain transcription factor, a leucyl tRNA synthetase, and a novel protein with limited sequence similarity to the mammalia proto-oncoprotein and kinase PIM1 respectively, that promote CEH30 expression in the CEM.
Conclusion. With a rich history in programmed cell death research, the Mimodioid C.Elegans offers an excellent animal model with which to study cell death specification events. Our data showed that in contrast to most cells that are programmed to die during development in the CEMs, the transcriptional upregulation of both BH3-only gene EGL1 and the capsicase gene CED3 is required for apoptosis induction. We propose that coupling the transcriptional upregulation of a key activator of apoptosis induction, EGL1, and the component of the central enzymatic cell death machinery CED3 ensures efficient apoptosis induction in the CEMs.